Hello, my name is Bria Fuller, and today we're going to be discussing the cell cycle. Now, what is the cell cycle exactly? Well, it's a complex set of stages that is highly regulated. The cell cycle is regulated by checkpoints that will ultimately determine the fate of the cell. The cycle is also directed by external and internal controls that will provide stop and go signs at the checkpoints. Now, let's go into a bit more detail about the cycle. The cell cycle consists of three distinct phases. There is interphase, the mitotic phase, as well as cytokinesis. Interphase consists of three phases, G1, S, and G2. During the G1 phase, the cell is metabolically active and continuously grows, but it does not replicate its DNA. G1 is followed by the S phase, also known as synthesis, during which DNA replication takes place. The completion of DNA synthesis is followed by the G2 phase, also known as GAP2, during which cell growth continues and proteins are synthesized in preparation for mitosis. Mitosis consists of 5 morphologically distinct phases: prophase, prometaphase, metaphase, anaphase, and telophase. Each phase involves characteristic steps in the process of chromosome alignment and separation. Once mitosis is complete, the entire cell divides in two by way of a process called cytokinesis. Prophase is the first stage in mitosis, occurring after the conclusion of the G2 portion of interphase. During prophase, the parent cell chromosomes, which are duplicated during S phase, condense and become thousands of times more compact than they were during interphase. Because each duplicated chromosome consists of two identical sister chromatids joined at a point called the centromere, these structures now, as you can see, they appear as X-shaped bodies when viewed under a microscope. The mitotic spindle also begins to develop during prophase. As the cell's two centrosomes move toward opposite poles, microtubules gradually assemble between them, forming the network that will later pull the duplicated chromosomes apart. When prophase is complete, the cell then enters into prometaphase, the second stage of mitosis. During prometaphase, the nuclear membrane begins to break down into numerous small vesicles. As a result, the spindle microtubules now have direct access to the genetic material of the cell. Eventually, the microtubules find their targets and connect to each chromosome at its kinetochore, a complex of proteins positioned at the centromere. As prometaphase ends and metaphase begins, chromosomes align along the cell's equator. Every chromosome has at least 2 microtubules extending from its kinetochore, with at least 1 microtubule connected to each pole. At this point, the tension within the cell becomes balanced, and the chromosomes no longer move back and forth. In addition, the spindle is now complete, and 3 groups of spindles are apparent. Metaphase leads to anaphase, during which each chromosome sister chromatid separate and move to opposite poles of the cell. Enzymatic breakdown of cohesion, which link the sisters together during prophase, causes the separation to occur. Upon separation, every chromatid becomes an independent chromosome. During telophase, the chromosomes arrive at the cell poles. The mitotic spindle disassembles, and the vesicles that contain fragments of the original nuclear membrane assemble around the 2 sets of chromosomes. Cytokinesis is the physical process that finally splits the parent cell into 2 identical daughter cells. During cytokinesis, the cell membrane pinches in at the cell equator, forming a cleft called the cleavage furrow, which is a band of microfilaments during cytokinesis and cell division that splits the 2 daughter cells' membranes in half. As I said in the beginning, the cell cycle is regulated by checkpoints that will ultimately determine the fate of the cell. A checkpoint can be defined as a control point where stop-and-go head signals can regulate the cycle. These signals will then report whether essential processes that should have occurred by that point have indeed occurred correctly and whether the cycle should continue or not. The checkpoints will also register signals from outside of the cell. The 3 major checkpoints in the cell cycle are G1, G2, and M. The G1 checkpoint determines whether all conditions are favorable for cell division to proceed in the cycle. External influences such as growth factors play a large role in carrying the cell past the G1 checkpoint. The cell will only pass the checkpoint if it is an appropriate size and has satisfactory energy reserves. At this point, the cell also checks for DNA damage. A cell that does not meet all the requirements will not progress to the S phase. The cell can sort of pause the cycle and attempt to cure the problematic condition, or the cell can advance into the G0 or inactive phase and await further signals when conditions improve. Then you have the G2 checkpoint, and the most important role of the G2 checkpoint is to ensure that all the chromosomes have been accurately replicated without mistakes or damages. If the checkpoint mechanisms detect problems with the DNA, the cell cycle is halted and the cell attempts to either complete DNA replication or repair the damaged DNA. The M checkpoint occurs near the end of the metaphase stage of mitosis. The M checkpoint is also known as the spindle checkpoint because it determines whether all of the sister chromatids are correctly attached to the spindle microtubules. Because the separation of the sister chromatids during anaphase is an irreversible step, the cycle will not proceed until the kinetochores of each pair of sister chromatids are firmly anchored to at least 2 spindle fibers rising from opposite poles of the cell. But what happens when the cell cycle is disrupted? DNA contains the hereditary material in nearly all organisms. 2 types of error or mutations can occur when DNA is improperly copied: silent mutations, which have no impact on the DNA sequence, and missense mutations, which change a DNA sequence. Missense mutations can multiply over time, leading to cell cycle disruption and formation of tumors, which are cells that do not stop dividing. Cancer occurs when the normal checkpoints regulating mitosis are overridden by a cancer cell, resulting in, as you can see, uncontrolled cell division. Cancer cells may not need growth factors to grow and divide, or they may make their own growth factor. They may even convey a growth factor signal without the growth factor being present. Cancer cells can go on dividing indefinitely if given a continual supply of nutrients. A normal cell is made cancerous by a process called transformation. Cancer cells will then form tumors, which are just masses of abnormal cells. If these abnormal cells remain at their original site, they are referred to as a bean tumor. However, malignant tumors metastasize, transferring cancer cells to other parts of the body where they can possibly form secondary tumors. In this diagram, you are looking at the formation of tumors. In this part right here, you can see that a tumor grows from a single cancer cell. Next, the cancer cells invade neighboring tissue. Then the cancer cells will spread or metastasize to other parts of the body. These cancer cells may survive and establish a new tumor in another part of the body. Now, let's talk about cyclins and cyclin-dependent kinases, which control the cell cycle. Cyclins are named such because they undergo a constant cycle of synthesis and degradation during cell division. When cyclins are synthesized, they act as an activating protein and bind to cyclin-dependent kinases, forming a cyclin-CDK complex. This complex then acts as a signal to the cell to pass to the next cell cycle phase. Eventually, the cyclin degrades, deactivating the CDK, thus signaling exit from a particular phase. A cyclin-dependent kinase is an enzyme that adds negatively charged phosphate groups to another molecule in a process called phosphorylation. Through phosphorylation, CDKs signal the cell that it is ready to pass into the next stage of the cell cycle. As their name suggests, cyclin-dependent protein kinases are dependent on cyclins, another class of regulatory proteins. Cyclins bind to CDKs, activating the CDKs to phosphorylate other molecules. As you can see in this graph right here, the concentrations of cyclin proteins change throughout the cell cycle. There is a direct correlation between cyclin accumulation and the 3 major cell cycle checkpoints. Also note the sharp decline of cyclin levels following each checkpoint, which show the transition between phases of the cell cycle, as cyclin is degraded by cytoplasmic enzymes. Now, let's talk about platelet-derived growth factor, or PDGF, which is one of the numerous growth factors, or proteins that regulate cell growth and division. PDGF is required for the division of fibroblasts, which are essential in wound healing. When injury occurs, platelets release PDGF. Fibroblasts are connective tissue cells which possess PDGF receptors on their plasma membranes. The binding of PDGF activates a single transduction pathway that leads to the proliferation of fibroblasts and the healing of a wound. Mitosis promoting factor is a protein complex responsible for triggering mitosis in somatic cells. Consisting of a cyclin B bound to a cyclin-dependent kinase, it catalyzes the phosphorylation of proteins that, in turn, bring about the events of mitosis, including condensation of chromosomes, formation of the mitotic spindle, and breakdown of the nuclear envelope. Levels of cyclins and MPF rise as the cell enters mitosis, reaching a peak during mitosis and then falling during anaphase. Now, that's it for the cell cycle. Thank you for watching.